Piezoelectric immunosensors. Piezoelectric immunosensors are those immunosensors in which we use an antigenic molecule and an piezoelectric material. Piezoelectric materials are unique type of materials. They have an ability to produce electric current when stress is applied on them. In piezoelectric immunobiosensor, the piezoelectric materials is used in the physical component of the biosensors. In immunobiosensors, the formation of antigen and antibody complex is the biorecognition process, and the biorecognition process is detected by a transducer in piezoelectric immunobiosensors. Normally, quartz crystals is used as a piezoelectric material in piezoelectric immunobiosensors. The formation of complex between an antibody and antigen is detected by the piezoelectric crystals. Like all other biosensors, the immobilization is the important step. The antibodies are immobilized on the piezoelectric sensing platforms. The piezoelectric sensing platform is needed to be sensitized or functionalized that the antigen or antibody could be immobilized on this surface. There is a variety of materials that is used for the immobilization of these biorecognition element. These materials are organic and inorganic in nature. The coating materials include silane reagent and modified silica and horseradish peroxidase based membranes, chitosan and multi-walled carbon nanotubes, zinc oxide micromechanical membranes, and protein A based films. They are used for covalent attachment to the piezoelectric crystal surface. So there are certain requirements for the attachment of these membranes. That these membranes should be thin, the coating should be homogeneous, they should be able to incorporate the amino acid functional groups, and they should contribute to the high sensitivity. Further, there should be minimum interference, and these membranes or the coatings should be able to produce the reproducible results. You can see in this slide there is a comparison between the two biosensors, in which in one piezoelectric biosensor there is no coating and the signals are very slim, but in the other case there is a surface modified protein film coating. As a result, it leads to an enhancement in the signals generated by the piezoelectric immunobiosensor. There are two types of technologies which are used in piezoelectric immunosensor. One of them is QCM technology. QCM is a label-free and extremely useful and sensitive device. We have studied about this device in our previous modules. Basically, what it does, it measures the resonance, and the mass is calculated depending upon the variation in the resonance. When there is an antigen antibody complex formation, there is a disturbance in the resonance, and this resonance is detected by the transducer. You can see in this slide. इस स्लाइड में आप देख सकते हैं कि एक क्वार्ट्ज क्रिस्टल पर प्रोटीन ए की मौजूदगी में आयरन नैनो पार्टिकल्स के साथ प्रोटीन्स को इमोबिलाइज किया गया. एंटीजन की इमोबिलाइजेशन के बाद सिग्नल्स की फ्रीक्वेंसी काफ़ी हाई थी, बट वंस इट डिटेक्ट्स द target molecule, which is shown in the red, there is the frequency of the oscillation is low. That is how we can use the piezoelectric crystals for detecting the target molecules in a piezoelectric immunobiosensor. The other technology is the use of micro cantilever. Cantilever is basically a structural element. It is a beam or a plate. It is attached at the one end and it is free at the other end. For the use in biosensors, cantilevers are commercially available. They are made up of different materials such as silica, silicon nitrides, different polymers, and quartz. Now you can see how this cantilever system works. There is an antibody immobilized on the cantilever beam. Once it detects the target molecule, there is the formation of an antigen and antibinding antibody complex. This complex leads to an increase in the weight, and once there is an increase in the mass, there is a change in the micro cantilever position. You can see a deflection, and that deflection could be detected optically. Now, in summary, we can say piezoelectric materials are very useful for the detection of target molecules in the piezoelectric immunosensors. Biological recognition element is coated on the membrane and that is attached on the piezoelectric material. Normally, quartz is used as piezoelectric materials. There are two types of technologies. One is QCM and other is cantilever technology that is used in the piezoelectric immunobiosensor. This method is very sensitive and very efficient for the detection of target molecules.